Hello and good morning, everyone. We are Group 7, and today we will be presenting our poster on Topic 4, which is caloric restriction, fatty acids, and the leptin-FTO axis, molecular insights into obesity and metabolic health.
My name is CHRISKAH and I will be presenting the background of our topic. First one is obesity background. Obesity is defined as excess body fat with a BMI of 30 or higher and is linked to serious health risks. Globally, adult obesity rates have doubled and childhood obesity has quadrupled since 1990. And in Malaysia, obesity rates continue to rise as part of the global nutrition transition. For the genetic factor, leptin is a hormone that regulates appetite and metabolism, such as abnormal levels are linked to obesity. Variants of the FTO genes like RS9939609 are associated with higher BMI, greater appetite for fatty foods, and an increased risk of obesity. These variants also raise the risk for the type 2 diabetes and metabolic syndrome. Last but not least, the dietary factors. High-fat diets contribute to excessive calorie intake and fat accumulation, which is fat provides 9 kilocalories per gram. Besides, the saturated and trans fats promote insulin resistance and visceral fat. However, calorie restriction improves metabolism, insulin sensitivity, and aids weight loss. Balancing fat quality and total calories is essential for preventing obesity and related disease.
Hello, my name is Claire Edward, and I will be presenting the problem statement, justification, and objectives of our study. For the problem statement, obesity is a major global health issue affecting more than 890 million adults worldwide. In Malaysia alone, over 54% of adults are either overweight or obese. Although diet and exercise are commonly recommended, many people still find it difficult to lose weight. This suggests that there are other factors such as genetics and hormonal regulation that are also involved. One key reason for this study is to understand how dietary fat and caloric restriction interact with the leptin hormone and the FTO gene, both of which play important roles in appetite control, fat storage, and energy balance. Some individuals gain weight more easily than others, even with similar diets, and this may be explained by these molecular mechanisms. By studying these interactions, we can gain insights that support the development of personalized nutrition strategies, which may be more effective in preventing and managing obesity, especially in individuals with genetic risk factors. The objective of our study is to explore how caloric intake and fat types influence leptin and FTO gene expression, identify how genetic predisposition and dietary factors interact at the molecular level, and finally evaluate how this knowledge can be applied in nutrigenomics and precision nutrition to improve obesity prevention and treatment.
Let's now explore how nutrients, hormones, and also genes interact to influence hunger, metabolism, and also obesity. First, leptin is a key hormone in appetite control. It's secreted by the fat cell and signals the hypothalamus in the brain about the body energy status. When the levels are high, leptin increases. This tells the brain to reduce hunger and use more energy. But during fasting, leptin drops, which increase the hunger and slow our metabolism. It's important to note that leptin helps regulate long-term body weight and not the short-term hunger we feel between meals. In obesity, leptin levels stay high, but the brain becomes resistant to it. This is called leptin resistant, where the brain stops responding to the leptin signal. As a result, the body keeps storing fat and burns less energy and stays hungry. Leptin also interacts with other hormones like insulin, cortisol, and also ghrelin, all of which affect appetite and fat storage. Now, let's talk about the FTO gene, short for fat mass and obesity associated gene. FTO regulates fat storage, appetite, and also metabolism. It produces an enzyme that modifies RNA to influence the genes involved in energy balance. When fasting, FTO expression tends to increase in the brain, triggering hunger. After eating, FTO levels drop to maintain the balance. Dietary fats and also affect leptin. Saturated fats, common in processed food, increase the leptin resistance and cause inflammation, leading to weight gain. On the other hand, unsaturated fats like those in nuts, fish and also olive oil improve leptin function and support the fat burning. Lastly, calorie restriction, or short as CR, affects both leptin and also FTO. When fat is lost, leptin drops, making the body hungrier and more energy conserving. FTO responds differently in different tissue, where CR might increase the FTO in the brain to stimulate hunger, but decrease it in the blood, which helps improve metabolic markers like lipids levels and also inflammation. Now, let's look at genetic risk factors, particularly the FTO gene, which one of the most studied genes linked to obesity. The FTO gene has been strongly associated with higher BMI and obesity risk. Key variants like RS9939609, RS8050136, and RS1421085 don't change the structure of FTO protein itself. Instead, they affect how nearby genes are expressed, especially those involved in fat storage and energy balance. People who carry the A allele of RS9939609 or the C allele of RS1421085 tend to feel hungrier, experience less satiety, and often prefer high-calorie foods. They also tend to consume more fats and carbs, not necessarily because they're less active, but due to how their metabolism responds. But here's where nutrigenomics becomes important. Lifestyle and diet still matter, even though for those with these genetic variants. For example, Ramadan fasting has been shown to lower FTO expression by 32% along with weight loss and also improve blood lipids. Another study also found that a high carb diet actually lowered the BMI in individuals with a certain FTO genotypes. Interestingly, fasting tends to reduce FTO levels in hypothalamus, while high-fat diet increase in fat and also the liver tissue. So, even though genetics set the baseline risk, behavior and also the environment shape the outcome. A balanced diet, regular physical activity, proper sleep, and also managing stress can all help reduce the risk. And don't forget environmental influence like food access, income level, and culture, and how walkable your neighborhood is. All of this can affect your weight regardless of genetics.
Today, I'll be presenting the role of nutrigenomic methods and how they are applied to better understand the link between diet and gene expression, especially in the context of obesity and metabolic health. Molecular nutrition is a fascinating area of research that focuses on how nutrients interact with our genes and how these interactions can influence health and disease. One of the key aspects in this field is gene expression, which refers to the process where genetic information is converted from DNA into RNA and ultimately into proteins that carry out specific function in our body. To study gene expression, researchers rely on various techniques. One of the most commonly used methods is quantitative PCR or qPCR. This technique allows scientists to measure the amount of specific mRNA, helping them understand which genes are active under certain dietary conditions. It's highly sensitive and accurate, making it ideal for exploring gene function. Another powerful technique is RNA sequencing, which provides a comprehensive view of all the RNA molecules present in a sample. Unlike traditional methods, RNA sequencing can capture both common and rare transcript, making it invaluable for identifying how diet can influence the entire transcriptome. DNA microarrays are also widely used, especially for analyzing the expression of thousands of genes at once. While newer technology like RNA sequencing are becoming more popular, microarray remain a cost-effective choice for large scales of studies. In addition to this, classical blotting techniques such as Northern blot for RNA, Western blot for protein, and Southern blot for DNA remain important for validating molecular findings. Meanwhile, CRISPR technology has revolutionized gene editing, enabling researchers to silence or modify specific genes to better understand their roles in nutrition and disease. Animal models play an essential role as well. For example, OB or oblique knockout mice, which have a mutation in the leptin gene, are commonly used to study obesity and metabolic disorder. These mice help researchers explore how diet and gene mutation interact to influence disease progression. Now, moving to the application side of nutrigenomics and nutrigenetics. One of the most compelling applications of this knowledge lie in understanding the FTO gene, which is strongly linked to obesity. Individuals with certain variants of this gene tend to have a higher tendency for fat storage and lower fat oxidation, especially when consuming high fat or high calorie diets. By identifying these genetic profiles, we can tailor diet plan that reduce fat intake and focus on improving metabolic health. Another exciting area is the role of epigenetic, which refers to changes in gene activity that do not involve changes in the DNA sequence itself. Caloric restriction, for example, can lead to beneficial epigenetic modification such as DNA methylation and histone changes. This modification can help silence harmful genes and may even serve as biomarkers to predict how someone might respond to a specific diet. All of these findings contribute to the growing field of precision nutrition, which aim to provide dietary recommendations that are personalized based on individuals' genetic and epigenetic background. This approach goes beyond general guidelines by taking into account how each person's body uniquely responds to food. In summary, the combination of advanced molecular tools and personalized applications is transforming how we understand nutrition and its impact on health. By integrating gene expression data, genetic variants, and dietary response, we can move towards more target, effective, and individualized nutrition strategy. That's all from me. Thank you.
Moving on to prevention strategies, there are a few diets mentioned, but the most appropriate one is Mediterranean diet, because Mediterranean diet is best suited for individuals with the FTO gene variant, because it promotes satiety and better appetite control. It is also a diet that is rich in monosaturated fats, fiber, and plant-based food, and it is also more sustainable and effective than low-fat or calorie-restricted diets. It also supports metabolic balance and reduces obesity risk. The second one is choose the right types of fats. Omega-3 fatty acids that can come from fish, flaxseed, walnuts. It improves insulin sensitivity, inflammation control, heart and metabolic health, and trans fat from processed or fried foods. It increases LDL cholesterol, inflammation, and disease risk. So, the FTO carriers should prioritize omega-3 and strictly avoid trans fats. And the third point is lifestyle intervention. It's a genotype-based plans which is improving the diet adherence, long-term behavior change, and cognitive and metabolic outcomes. And best results seen with combined strategies like diet plus exercise plus brain training plus stress control, and genetic insight enhance motivation and supports personalized preventions. Lifestyle choices can influence gene expression via epigenetics. Current treatment and management for obesity. There are two categories, which is non-medical treatment and medical treatment. So, for non-medical treatment,
The first one is lifestyle and community support. Eat fewer calories, be more active, and change eating habits. Plans should match each person's culture, needs, and feelings. Programs like COSPAN help through peer support and early health checks. The second one is digital tools. Use apps and online platforms to track food, get coaching, and stay on track. It is very helpful for people in rural areas or with busy lives. The third one is diets for FTO gene, which is a gene for obesity risk. Intermittent fasting (IF) reduces hunger hormone, leptin, and may help FTO carrier the most. Ketogenic diet, aka keto, may improve hormone balance and reduce cravings. Low-carb diets results vary. It works better for people that is younger or over longer time. Next is medical treatment. The first one is medications. Drugs like semaglutide help reduce hunger and support weight loss. People with the FTO gene may respond less due to how their body works, handles hunger signals. Other genes can affect how well the medicine works and also the side effects. The second one is weight loss surgery. It is best for severe obesity when the other methods doesn't work. It includes sleeve gastrectomy and gastric bypass. FTO carriers may lose a bit of weight, but will still benefit for it.
Now, let's move on about the controversy related to this topic. First, unclear role of FTO gene. The FTO gene has been linked to traits like appetite and fat metabolism. A study by Ponce Gonzalez found that people with the TT genotype felt hungrier after exercise but also burned fat more efficiently. This suggests that different FTO genotype may affect how individuals respond to exercise and diet. However, other studies have found contradictory results linking to the ALL instead with increased body fat and appetite. This inconsistency makes it difficult to confidently use FTO data in designing personalized diet plans. So, further research is needed before it can be applied in nutrition or public health settings. Now, moving to leptin and its link with obesity-related hypertension. According to Au et al. In 2022, leptin levels are strongly associated with body fat, but not directly with high blood pressure. Their findings show that people with both obesity and hypertension had leptin levels similar to those who were just obese, which suggests leptin may not be the main cause of hypertension. This challenges the idea of targeting leptin or FTO alone through diet and raises doubts about how much dietary fat intake or calorie restriction can actually influence this pathway in managing obesity or blood pressure. Let's look at the ethical, legal, and social issues surrounding the use of genetic information like leptin and FTO in obesity and diet planning. Ethically, there is limited scientific proof that using genes like FTO or leptin can effectively guide personalized diet. Recommending diets based on unproven data can mislead people and take focus away from lifestyle habits that we know work. Informed consent is also important. Patients need to understand the risks, limits, and emotional impact of testing. Legally, many countries lack proper rules of direct-to-consumer genetics testing. This can lead to misleading health claims. There's also the risk of genetic discrimination, especially in jobs or insurance. While countries like the US and Canada have laws to protect people, many others don't. Socially, Access to testing and personalized diet is often limited to higher income or urban population. These create health gaps between communities. Also, focusing too much on genes can lead to stigma or make people feel helpless. On the other hand, blaming only behavior ignores bigger issues like poverty or food insecurities.
In conclusion, this report examined how dietary fats, caloric restriction, and the leptin-FTO axis are connected to obesity and metabolic health. We found that obesity is not just influenced by lifestyle, but also by genetic factors, making it more complex than it seems. Research shows that consuming too much saturated fat and calories can lead to leptin resistance and increased FTO gene activity, both of which contribute to weight gain. On the other hand, caloric restriction and healthier fats such as omega-3s can improve metabolic outcomes, especially in people with certain FTO gene variants. We also discussed several scientific tools such as qPCR, RNA sequencing, SNP genotyping, and CRISPR, which allow researchers to study how genes and nutrients interact. These tools are essential for nutrigenomics and precision nutrition, which aim to develop personalized diets based on a person's genetic profile. However, there are still some challenges, including inconsistent findings, ethical concerns, and limited access to genetic testing. So while personalized nutrition holds great potential, it must be applied with care and responsibility. So that's all from our group. Thank you for listening.